particular interest in our population. She's done quite a bit of research in visual processing after resections. And her student, who is now in medical school, has also really taken an interest in our population. So as he goes through medical school, you're all going to be top of mind for him. I'm particularly fond of you, Michael, because you do everything I ask you to do, which is great, which keeps him in the loop. So yes, thank you so much.
Okay, hi everybody. For those of you who don't know me, my name is Michael Granovetter. As Monica said, I'm a medical student in the MD PhD program at the University of Pittsburgh and Carnegie Mellon. And today I'm really excited to get to share and talk about some work that really wouldn't have been possible without many folks in this room and the Brain Recovery Project as a whole. And that work is going to focus on how does childhood hemispherectomy affect visual recognition. Now, throughout this talk, I'm going to be using the terminology hemispherectomy as kind of a catch-all umbrella to include hemispherectomy surgeries and hemispherotomy surgeries, as well as hemidecortication. And what we're going to be focusing on today is the visual system. So I want to give a brief overview of vision in the brain just to get us started. So let's talk about what we already know about how hemispherectomy can affect vision. So anybody who, like myself, flew into the Salt Lake City airport might have seen this terminal. And how we visually process this terminal and this viewpoint is the left side of your visual world goes to the right side of your brain. And the right side of your visual world goes to the left side of your brain. And together it kind of makes a complete picture for all of us. And as some of you might be familiar with, after a hemispherectomy, for instance, to the left side of the brain, you lose this part of your visual field. So now you're only getting information from the left side of your vision, and it's going to the remaining hemisphere that's left, the right hemisphere. Now, this isn't to say that this is always a detrimental day-to-day impairment for individuals with what's called a visual field cut or hemianopia. Because individuals with hemianopia are able to kind of adjust their gaze so that they're taking in as much information from the visual world that they can. And in some cases, really can't even notice that they have the hemianopia or the visual field cut unless there's something very specifically close kind of in their right visual field. They're always readjusting their gaze to try to compensate in some way. And what we know about visual field cuts after hemispherectomy is that these are pretty much permanent. So once you lose one side of the brain, you're not able to recover function to allow you to be able to see in your whole visual field, even if you're able to make other sorts of adaptations to allow you to move your eyes around to take in the full picture of what you're seeing in your world. So we already have a lot of evidence to understand that part of the visual system following hemispherectomy. But vision is so much more than looking at a picture in your visual world. We have to take that information and process it. So say you're in the airport terminal and this is your friend who's picking you up from the airport. You need to be able to not just see them, but be able to recognize their face, recognize who they are. Or you might need to read words. Word reading is something that requires vision. You might need to be able to know which gate that you want to go to. So that's what I mean when I say visual recognition. It's more than just the information that you're seeing. It's how are you processing it? Are you able to, say, distinguish the face of your friend from your mailman, or be able to parse apart words to allow you to read appropriately? And so the focus today is going to be on faces and words. And the reason I'm going to be focusing on faces and words is we know quite a lot already about how faces and words are organized inside the brain over typical healthy child development. And let me illustrate that for you. So when you're a child, when you're first born, you don't know how to read quite yet, but you're constantly being exposed to faces in your environment. And faces can be processed on both sides of the brain, both your left side of your brain and the right side of your brain. But then kids eventually get older and they might go to school and they'll learn how to read. And some of you might be familiar with the fact that language systems in the brain are on the left side. I always mix up my lefts and rights, so the way to keep that straight is left for language, L, L. They both start with L. And so because language is typically on the left side of the brain in the majority of the population, not everybody, but in most individuals, your ability to process and recognize words starts to develop its function in the left side of the brain as well. But that's where your brain is already trying to recognize and process faces. So what happens over time is that even though you might have, say, a little bit of ability to process words in the right hemisphere, it mostly happens in the left. And by the time you reach adulthood, words are mostly on the left in terms of function, and faces are mostly on the right, even though each hemisphere can do a tiny bit of both. And we know this from lots of studies where we've imaged healthy adults under MRI. So this demonstrates that this is brain activity in adults who had MRI. And you can see here in purple, brain activation when the individuals inside the MRI were seeing letter strings on the left. And you can see more brain activation to faces here in orange on the right. So again, you see mostly words on the left, mostly faces on the right, a little bit of orange and pink on both sides of the brain. But you see kind of the segregation of function in adults. And so that kind of raises the question, well, so does that mean if these functions become segregated that we need both hemispheres in order to engage in visual recognition? Data from adult patients would seem to suggest so. So oftentimes, if an adult has a stroke that affects even just a tiny part of the visual system on the left side, some adults might end up having a word reading impairment. And other adults, like this individual who had a stroke on the right side of their brain, might suffer from something called face blindness, or you might hear the term prosopagnosia, the inability to distinguish between different faces. But again, this is in adults. We're not born necessarily with words on the left and faces on the right. This is an organization of the brain that starts to emerge over childhood. So that raises the question, in the case of removing an entire hemisphere during childhood, before you see this complete segregation of function that you see in adults, can an individual who has, say, a left-sided hemispherectomy or hemispherotomy, or a right-sided hemispherectomy or hemispherotomy, be able to maintain function for both word and face recognition? And that's what we set out to do in this particular study, to essentially ask the question, is development with just one hemisphere enough to be able to engage in both face and word recognition processes? And really crucially, does it matter which hemisphere is removed? Is it going to be the case that we might predict based on what we saw in adult stroke patients that if you remove the right hemisphere, is that going to affect your face recognition more? If you move the left hemisphere, is that going to affect your word recognition more? Or is it possible that with whatever hemisphere is left behind, I think my laser pointer is dying here, but, oh, sorry. With whatever hemisphere is left behind, can you have function for both faces and words, whether it's on your right side or on your left side? So I was really fortunate to get to work with many, many individuals from the Brain Recovery Project who we are incredibly grateful for. About 24 who have had left-sided surgery and 15 who have had right-sided surgery. And we had all these individuals participate in tests at the Brain Recovery Project conference in Cleveland in 2019, as well as online over Zoom. And what did these individuals do? So we tested them with very simple tests on face and word recognition. Essentially, we had individuals see one face very quickly and then another. And all they had to say was, were these the same or different? And then we did the same thing with words. So they would see a word like soap, and then a word like soup, flashed really, really quickly. Like we're talking about less than a second, a fraction of a second, to make this task difficult enough for us to be able to see potential differences between patients. And we also tested a group of individuals who had never gone to surgery. And what did we find? There's a lot of information on this graph, so let me just quickly walk you through it. So here on the x-axis, we have our different groups of participants. We have what I'm calling controls. These are individuals who did not have brain surgery. And we have folks who had a left hemispherectomy and folks who had a right hemispherectomy. All the individuals who have had hemispherectomies are in red. All the controls are in blue. And we tested them on both accuracy for faces and accuracy for words. Now you might look at this bar graph and say, okay, hemispherectomy participants had lower accuracy than the control participants on both tasks. You can see that difference here. Here are the controls in blue, and then here's the average of the individuals with hemispherectomies in red. But I want to point out two very important things here. This dotted line is showing what you would see if an individual was pressing buttons completely randomly, that they weren't able to recognize faces or they weren't able to recognize words. Patients actually perform quite well on this task at over 80% accuracy for both words and faces. They do quite remarkably. And most importantly, there were no differences between the left and right surgery participants. It's not that if you have surgery to one side or surgery to the other, you're going to have more of a deficit in faces or more of a deficit in words. You don't see the sort of dramatic impairments that you might see in adult individuals. Both groups are performing quite well at above 80% on average. So that would seem to suggest that if you have a hemispherectomy and you're left with just a left or right hemisphere, each hemisphere of the brain is able to engage in face and word recognition. A little bit lower than if you didn't have brain surgery, but still remarkably well considering how much brain is removed. And the real question is, here we're testing behavior. Can we actually look in the brain and see what these individuals' brains look like compared to kids who have never gone to surgery? So we can do that with MRI. And we actually had folks recruited in Pittsburgh, but we also had a number of people that we met through the Brain Recovery Project actually fly to Pittsburgh to participate in our MRI experiments. And what we did is we had 9 individuals, about 5 of them had full hemispherectomies, and 4 had smaller surgeries, but that still affected the visual system. And we compared their brain activity to 21 healthy kids. And what we found were 2 things. First, while individuals were in the MRI, we showed them different sorts of pictures. For instance, faces. And you can see here in red, these are the areas that respond to faces. You can also see here in turquoise, here's the area that responded to words. And here in dark blue are the areas of the brain that respond to visual inanimate objects. And what we actually found was that the amount of brain that responds to these different pictures is actually very similar to what we see in kids who have never gone to brain surgery. So if we look at the right hemisphere of this example patient, on average, their amount of brain responding to these pictures is pretty similar to what you would expect in the right hemisphere of somebody who didn't go to surgery. And what's more interesting is the fact that the brain activity processes that are somewhat involved in actual visual recognition are also similar between individuals who had hemispherectomies or large surgeries and healthy individuals. So in the real world, we have to be able to distinguish and say, hey, you know, is this one of my parents? Is this my boss, et cetera, et cetera. And there are particular brain activity patterns that allow us to capture how well is the brain potentially doing this. Same thing for being able to distinguish between different words. Again, this was similar between individuals who have had these brain surgeries and healthy individuals. And there's more that we can do with brain imaging. Dr. Behrmann is going to talk about even more in-depth imaging studies that we did. But I want to end here to say that what I've been able to show with the behavior data I showed you, the MRI data that I've showed you, is I've partially answered these questions. Is development with one hemisphere enough to be able to engage in these different visual processes? And do we need both hemispheres? The answer is kind of a yes and a no. So remember, the individuals with hemispherectomies had about, on average, 80 to 90% accuracy. Individuals who didn't go to surgery had a little bit above 90% accuracy. But it's really important to take away from this that 50% loss of brain isn't equivalent to 50% loss of function. You maintain way more function than you might otherwise predict. And so, unlike in the case of an adult who loses, say, the right side of the brain and might become face blind, what we see in kids who have surgery while they're potentially still developing, we see the ability of one hemisphere to be able to engage in multiple visual processes. And that can occur whether the surgery is on one side or the other. So what's next? Well, there's still so much work that needs to be done to be able to provide clearer answers for families about what we know about brain recovery. For instance, I've demonstrated how the brain is able to, after surgery, support face and word recognition, which we know are typically in adults on opposite sides of the brain. But there are many, many other brain functions as well that might have that split. How do these results translate to other abilities and other functions? Also, you might be wondering, well, if my child has had surgery, how has that affected their ability to process words or their ability to process faces? And even though we were lucky enough to be able to work with a group of 39 individuals with these surgeries, it still wasn't enough for us to be able to make conclusions about how we can predict outcomes. There was a lot of variability. Some individuals performed above the average of all of the hemispherectomy patients, above 90%. Some performed a little bit below the average. There was a big range in performance, even though the average is 80. And what we're hoping to do moving forward is the more data that we're able to collect, the more we're going to be able to learn about how does age at surgery affect outcome? How does the reason for getting surgery, the different sort of etiologies of disease, how does that affect outcome? And that's still a mountain that we still need to climb. So with that, I'd like to just throw out a couple of acknowledgments to my mentors, Dr. Marlene Behrmann and Christina Patterson, who oversees the pediatric epilepsy surgery program in the Department of Child Neurology. Individuals who have helped in this work, Sophia Robert, Leah Edinson, Dr. Mallow, and Dr. Freud. A huge shout out to Monica Jones and everyone at the Brain Recovery Project, including individuals in this room who we really owe a lot of gratitude to. And again, that's you guys. And these are my funding sources. And please definitely reach out to me if you have any questions. Thank you. And I think there'll be time for Q&A after Dr. Behrmann speaks, is my understanding.
Okay, my name's Marlene Behrmann. I am in the Department of Ophthalmology at the University of Pittsburgh and also at Carnegie Mellon University. This is my email. You can just Google me, you'll find me. I'm happy for you to be in contact with me. I want to thank you for the opportunity to talk to you today and to present some of our research findings. I will mostly be talking about the status of the visual system in the brain after surgery, but we are also currently running studies that look at changes in the visual system from pre-surgery to post-surgery so that we can understand the entire scope of visual function. Before I do anything, I need to express gratitude to people. Firstly, all of you, the families and their children. I can see some families here whom I know who flew to Pittsburgh for these studies. I have a great debt of gratitude to Monica Jones, whom I'm sure you all know is a force of nature, and it's been my true privilege to cross paths with her. And the many people who make up the BRP organization, the volunteers, etc., etc. All of these people have given us the unprecedented opportunity to examine the visual system in the brain and to shed light on the kinds of changes that might be seen in individuals with hemispherectomy. Okay, this work would not have been possible without the huge amount of work and dedication of my students, Michael Granovetter, who you've just seen, Sophie Robert, Maria Kronios, and Tina Liu. And if any of your family has participated in the research that we're doing here, you will have encountered one of them. They are working 12 hours a day to run the experiments here. I've also have postdocs in my lab, and I'd also like to recognize my collaborators, Dr. David Plaut, Dr. Christina Patterson from Children's Hospital in Pittsburgh, and Dr. Taylor Abel from neurosurgery. Some of you may have heard him speak earlier in the day. And in fact, Dr. Abel introduced me to Monica Jones, so special thanks to him. Okay, so I'm not going to give you an anatomy lesson or a physiology lesson, but I really do need you to understand a few things about the visual system of the brain. So just like stick with me on this journey. This is the human brain. You're looking at it from the back. The pink area here is the occipital lobe. The green area is the temporal lobe in the left hemisphere, and this is the one from the right hemisphere. And if you turn the brain over and look at the base, like if I took wire cutters and cut my head off and turned it over, you'd be looking at the occipital lobe and the temporal lobe. And these are the primary regions that support visual function. A little bit also parietal cortex, but we're mostly talking occipital temporal cortex. Not only are there many, many regions, like 150 regions in one hemisphere, there is also massive amounts of communication between all of these areas, sending signals forward and backward to provide this rich and meaningful, rapid understanding of our visual world. I don't know about you, I open my eyes and everybody looks so clear. It's why we thought that you had a camera in your head, because it feels like a camera. It's so, it's effortlessly depicted and rich. Okay, and that is because there are lots of areas and they are sending signals very rapidly. The first kind of question we want to ask is, all right, there are all these regions. Which regions respond to which stimuli? So I'm going to build a little bit on the kinds of studies that Michael already described. We have participants in the magnet, and there is a screen inside the ball, and we show pictures of words, faces, objects, places, and bodies. And there is a very typical result that we see. So I'm going to show you the results from the base of the brain, and it looks as follows. Okay, the eyes are over here. This is the occipital lobe at the back here, and this is the temporal lobe. Oh, the one thing I really need to explain to you is that the right hemisphere is almost always shown on the left in terms of radiological conventions. This is sort of the medical standard. And it's a little bit like as if I were looking at you. So what's on your right side is on my left side. So I will always label the hemispheres because it's super confusing. And this is what we see. There is a lot of area of the brain that is involved in recognizing objects. There are areas in green here that are involved in recognizing places, buildings and landscapes. A small little body area here. And then areas in both the right hemisphere and the left hemisphere associated with face recognition. Shows you how important faces are, that we have it in both hemispheres. And lastly, an area in the left hemisphere for recognizing words, again, reiterating what Michael said, and that is that it's in the left hemisphere to connect up with language areas that are also in the left hemisphere. Okay, I'm going to refer to this kind of picture as the visual map. Okay? And the question is, what does the visual map look like in individuals who've had parts of the visual system resected? So it could be an entire hemispherectomy. That hemisphere's visual system is gone. But it could also be a more, these actually all, this is, you can see here, this is an anterior temporal lobe resection, which is also part of the visual system. So in our early studies, because we know extremely little about this visual map, we had participants with hemispherectomy, hemispherotomy, just a lobar removal, or even just a laser ablation, so that we could begin to understand the landscape of the visual map. So the individuals with occipital and temporal resection, again, either from hemispherectomy, urotomy, lobar, and some parietal, are going to be our test group, because in those individuals, the visual system has been surgically removed. We're going to have a control group. These are kids who are the same age as our test group, who allow us to compare what typically developing visual maps look like, so that we can then benchmark the maps of the patients. And we want to ask questions like, is the visual map shifted at all? To the extent that it remains the same, does it show the normal kind of activation? In other words, is the code that the visual system is using in individuals following surgery the same as in typical kids? Or is it maybe just working a little bit differently? Lots and lots of questions. There are only a handful of studies in the literature. I'm going to share with you really just a small subset of the studies we have done, and then I'll tell you a little bit about what we are going to be doing going forward. Okay, so why should you care about this? Well, many of you know that your family member is hemianopic or hemianopsic, and so you have first-hand experience of what it means to have a visual deficit. We don't know enough about the visual system, and so the reason that I care so much is, in order to be able to think about targeted intervention, we first need to understand how the system works. We need to map out the system, and we need to understand what it looks like so that we can intervene, and we don't yet know what it looks like. Okay, Michael already mentioned to you that we have only seen persistent hemianopia or hemianopsia. We have not seen recovery from hemianopia. So this is just a quick example. This is a young boy who had an occipitotemporal lobectomy at age 6. You can see his tumor over here. This is the resection. And this is my way of showing you how much of the right hemisphere was removed. This is his ophthalmological examination showing that information in the left visual field, which is in black over here, he does not respond to any visual information shown to the left. And every time that we test these kids, we do that same kind of experiment. So this kid, we saw at age 7 years, 10 months, and again at 10 years, 10 months, and we redo that kind of characterization of the visual field loss. Red here indicates that there was no response to stimulation on that side. And this was true when he was 7, and this is true when he was 10, and every time we've seen him since then. The good news, though, is that there is general consensus in the field, and that is despite the homonymous hemianopia, the majority of post-surgical children have good visual outcomes. And that is awesome. Michael showed you some of that already, that these kids are performing at 80%, which is actually really quite good, given that they have about 50% of their brain. So, I want to show you how we do these studies, not just the results. So this is a patient. This is his initials. It's a code. We never give information about the identity. It's all confidential. It's all coded. So this individual with, as you can see here, an anterior temporal lobe resection, lay in the magnet. We show these kinds of stimuli. And then afterwards, we can kind of put a pin in as to exactly which areas are activated by faces. And you can see here that both in the right and the left hemisphere, there is face activation. And we can put another pin in for the areas that are activated by words. And you can see here, the left hemisphere has word activation, but the right hemisphere does not. So this is like putting a pin in on a map. And that's exactly what we've done here. And then we can transfer those locations of activation onto a kind of a map. It's like an atlas of each individual child's brain. This is the left hemisphere, this is the right hemisphere, this is how far anterior, forward, and this is towards the back of the brain. And so we have these pins for each of their visual functions. And we can do this individually for every single participant. And then for every participant, we transfer it onto this little atlas, left to right and front to back. And we can examine how well their visual map compares to the matched control. So this is what it looks like in typically developing kids. And here are the sort of atlases for each of the individual children. Now, some of them, like this one, has got pins for every single function, and it does not differ at all from the controls. Notice the next one, where we only see pins in the right hemisphere, and there is no activation on the left-hand side. But nevertheless, the activation that we see in the right hemisphere is the same as the control's right hemisphere. And so the conclusion from the study, and I think we had something like 20 kids in the study, I'm just showing you about 6 here, is that whatever part of the brain remains, in the majority of cases, we see the correct visual map. Like things haven't gone completely haywire. And there is also a normal strength of response. It's like that area of the brain is actually doing what it ought to be doing. So, the first take-home message is, if the tissue is preserved, we get normal, sort of typical, we get a typical pattern of activation. So that's all well and good, but I'd like to take a deeper dive with you. And that is, as Michael mentioned, and as I will go into further detail, there are differences between the hemispheres. So faces activate both the right and the left hemisphere, and words activate just the left hemisphere. So what does the visual map look like in an individual who has only one visual cortex? Okay, and moreover, when we see these patterns of brain activation, sort of the neural signature, can we plot the changes in the visual map over time? Can we observe plasticity at work? What I showed you previously was we scanned the kids one time, whatever age they were, and we looked at their visual maps. Here we are going to do longitudinal studies that allow us to track and monitor the changes in the visual map as kids mature after surgery. There are, in fact, people use the term plasticity very often and somewhat loosely, I dare say, because we don't really know that the changes that we're seeing is because the brain is plastic. I want to show you that if we do the studies, we can actually plot out the profile of plasticity. And that's really important. We want to understand what the capabilities of the brain are post-surgery. Okay, these areas probably look extremely small to you. It's like maybe you're even peering to see them. I want to tell you that even though they look tiny, each of these areas contains something like 1 million nerve cells, 1 million neurons in each of these areas. Okay, so I'm first going to show you the same individual who I showed you his hemianopia in the very beginning. So he's got a posterior occipitotemporal resection. He had a tumor was detected at age 4, surgery at age 6 years and 9 months, and then we scan him every 6 months after that, and we can zero in specifically to look at the plasticity of the area in which we normally see the visual map. Okay, so it's this area over here, and I'm showing you in 3D which particular part of the brain we're looking at. This is an illustration of this area in 3D. And the redder each little bit of this is, it means the more those nerve cells are biased for face recognition. And the more blue it is, you can see the legend over here, the stronger the blue, it means that little bit of the brain is now committed to word recognition. So when he was young, actually there's not much commitment here. There's sort of pale blue, pale kind of, there's not much red going on. But as we continued to scan him, you can see eventually we get much stronger red and much stronger blue. Actually this, I apologize, I think that the resolution here is not very good, and the pictures are not as beautiful as they ought to be, so please bear with me. But I assume you can see that there is sort of strength emerging over time and commitment to face recognition or word recognition. So that's great. It's great to see the emergence of both faces and words in the left hemisphere after a right resection. But you may be thinking, or you should be thinking, well, that's not very exciting because we already have face and word recognition in the left hemisphere in everyone. So, okay, so you see it in these kids. I mean, that's nice, but it's not dramatic. So hold on for the drama. Okay. What happens if the left hemisphere is resected? How well does the right hemisphere visual system do on its own? So this is a kid who has had not a full hemispherectomy, a posterior, parietal, occipital, and some temporal lobe resection, but these are the visual areas that we're interested in, so it's okay for the study. Her first seizure was at age 7, surgery was age 13, which is a little older than what we typically see. And we also scan her over time. And I'm just going to show you two of the time points. Again, the images are not very precise. I hope you can see that from the first scan about age 13 and a half to age 15, we see this increase in the organization of the visual map, with some areas kind of taking on the responsibility of face recognition and neighboring areas taking on the responsibility for word recognition. Okay, so we're able to monitor the plasticity, and it's really dramatic. Word recognition does not typically appear in the right hemisphere, and yet, if your left hemisphere, occipitoparietal temporal region is missing, the right hemisphere sort of has no option but to take on the task, not only of face recognition, but making room for word recognition as well. And it is possible to see the plasticity, to see the emergence of word recognition in the unusual right hemisphere configuration. Okay, so mostly what I've done so far is tell you about all these brain areas, the visual map this way and the visual map that way. Of course, all of these areas are deeply interconnected with each other so that they can communicate very rapidly, send neural signals from one region to the other. The regions talk to each other. And so I'm going to just show you a tiny little bit about the work that we've done on measuring the strength of the connections between all of these areas. And I'm going to show you some work that looks at the structural connections, I'll describe it to you, and then the functional connections. Okay, so these are a different kind of brain scan. This is from this paper, but we do exactly the same studies. I'm going to be interested in these two tracts. Please don't worry about the actual names. What's important here is that both of them start in the occipital lobe and go all the way through the temporal lobe, and in this case, terminate in the very anterior part of the temporal lobe. Or for this tract, it goes from the occipital lobe, partway down the temporal lobe, and then it swings up into frontal cortex. This is the same tract projected onto the MRI scan. This is just like an illustration, and that's sort of the real thing. So we've been looking at these, I'll call them fiber tracts, these white matter. It's kind of like the wiring cables, if you think, electricity or circuit or something. So there are these two tracts I told you about. Here one is purple, one is green. These are data from one of these control participants, an 11-year-old male. The left hemisphere's got two tracts, the right hemisphere's got its two tracts. Everything looks fine. We can definitely get this kind of result repeatedly and robustly. So now we start looking at what happens to individuals with right resections and left resections, and if the resection is early or the resection is late. And we have actually observed several different patterns. So let me give you some examples. This person, who you can see has just got a left temporal lobe resection, doesn't have any tracts in that temporal lobe, which is a little surprising. The resection is quite focal. It's just like a specific region, just like the most anterior pole of the temporal lobe. And yet it seems like all the tracts in the left hemisphere have sort of degenerated. It's uncommon. We've only seen it in this individual, but that is sort of the extreme, one extreme. We see kids like this who show both tracts. This individual, even in the resected hemisphere, is showing both tracts, and they are running very close to the midline rather than in the sort of lateral section that's being resected. And many other types of patterns as well. We don't know yet why these different patterns emerge, and we don't know what these different patterns mean. And we're beginning to understand, though, what these different patterns imply in terms of preserved visual function. So we're very much in the midst of kind of exploring these kinds of questions. But regions of the brain that don't have these real cables between them can also communicate with each other. And I will call that functional connections rather than physical connections. So again, there are very, very few studies in the field. Dr. Lynn Paul, who is here, and her colleagues published one paper on functional connections in adults, and that was extremely useful. What we wanted to do here is both look at the functional connections in children, but we also wanted to look not just in the visual system, because of course the visual system is part of the whole brain. And so we're going to look not just in the posterior part where I focused all my attention up until now. We are going to look at the entire brain. And the way this is work that's ongoing, and the way in which we do it is the kids lie in the magnet and we show them a movie. This is the movie. It's called Escape to Witch Mountain. Maybe you know it. It's kind of an age-appropriate movie for young kids. There's like no violence, no gore. It's just like a really nice movie. And then in every little region of the brain, we look at how much activation there is over the entire time course of the 11-minute segment of the movie. And you can see this is the tip of the occipital lobe, and it has this pretty nice pattern. When new exciting things come on the screen, this part of the brain sort of gets even more activated. We have lots of kids, hemispherectomies on the right and on the left. And we are going to now examine sort of the communication between all of these regions in the preserved hemisphere. This work is very much underway. We don't have the complete results just yet. Don't worry about all the equations here. Essentially what we're doing is looking at the communication profile, let's say, of this most posterior region with this region next to it, with further regions, with even further regions. So we're looking at the activation pattern between even remote regions and seeing how well they communicate with each other. Okay, so I've told you about the typical organization in whatever brain tissue is preserved. I've told you that we have had this really exciting opportunity to watch the visual system unfolding over the course of maturation and development in kids who've had resections of the visual system. And I've told you that we are now making headway in understanding the communication between different areas of the brain and potentially strengthening between these different areas, or even possibly weakening in kids who've had resections of the visual system. We're collecting data here at the BRP. Some of your kids or you might have participated. We're pushing beyond what we've already done and asking two new kinds of questions. One of them has to do with how the eye, the nature and extent of the coordination between the two eyes in individuals with a single hemisphere. So normally if you've got two hemispheres, it can kind of coordinate your eye movement, and your eyes work together. It's actually sort of phenomenal. There's like this sort of such a close partnership between the two hemispheres. Your eyes move together, unless somebody is squint and one eye goes in a different direction. And so we are trying to understand how this is possible in somebody with just a single hemisphere. Like, how does this happen? So we brought an eye tracker with us. TSA were really unhappy with me. They made me unpack everything. It looks like a torture device. But have seen me on the floor. Yeah, could have seen me on the floor in the airport in Pittsburgh. They took my entire suitcase apart. I seem to have lost, my toothpaste on the way and all kinds of things. But we did bring it, and we actually have already, since Thursday, collected more eye movement data than already exists in the literature to date. So I hope that the next time we meet, I will be able to tell you much more about the eye movements. The other direction that we're pushing in has to do with attention. So even when the visual information goes into the brain, it's really important when we look at such a complex visual world that we attend to particular parts that are of greater interest to us. Otherwise, you become so distracted and sort of overwhelmed by all this input. So we want to understand now, how does the attention system work in individuals with a single hemisphere? And this is the second set of experiments that we're running here. Again, this is the good news and the bad news. The good news from a scientific point of view is that there are no studies. Almost whatever we do is going to be a major contribution. The bad news is there is no precedence, there isn't an existing understanding. And so we're trying to break new ground, and we're sort of hoping that we're going in the right direction. That's sort of the pros and cons of the science. Okay, so this brings me to the end of my talk. Again, it's because of you that we have this unprecedented opportunity to learn about the visual system in general, about the visual system in kids with a single hemisphere. And only when we have laid these foundations can we begin to think, how might we be able to promote change in the visual system? How could we enhance visual function in these kids? So thanks to the families and the children, Monica Jones and so many others, the whole BRP organization, my students, postdocs and collaborators, Michael Granovetter, who you just heard, shouldered the majority of the burden for our testing at BRP and for much of the work in my lab, and my two more junior students, Sophie Robert and Maria Kronios. And this is my email, but you can also get me Behrmann@cmu.edu. You can Google me. I am happy to receive email from you. I'm quite good at answering. I assure you I don't have the answers, but I'm happy to answer. Thanks very much. I think Michael and I are both happy to take questions. Do you want to stand up, Michael? There was I know there was a question from before that we put off.
Yeah, in the study that was done with the 39 different subjects.
Yeah.
What was the median age of the kids in there?
That's a great question. It was about median age at participation was, I can't give you the exact number, but it was somewhere between 15 and 17.
I would say 15.
And that's the median age at participation. Almost everybody in that study had the hemispherectomy as a child. So it was childhood hemispherectomy, but we got to see folks at different ages. And one thing that would be really nice, and that's why we love to recruit more and more folks to participate, is being able to tease apart, age at the time that they participate versus age at the time they had surgery. We need more data on that.
And age at the time of first seizure.
Yes.
So the more people we have, the more we can begin to understand how these different factors play into the outcome and the prognosis. Go ahead, at the back.
Yeah, that was a question I was going to ask, and maybe this is, you don't have the answer, but with word recognition and left hemisphere, is there a difference between kids who sort of develop that word recognition, in the left hemisphere before left hemisphere surgery, versus kids who, my daughter age 4 at the time of surgery never really recognized words before that part of the brain.
Yeah, it's a super question, and it's a very good question to ask. So if a kid, let's assume that this kid has got language in the left hemisphere, like most of us here. and the kid was about, I don't know, 6 or 7, started going to school, started learning to read, and the word area is developing in their left hemisphere. And then they go to surgery, and the left hemisphere is resected. Now what happens? We have seen, in fact, the kid who I showed, who'd had a left resection and developed the word area in the right hemisphere, was 13 years old at surgery. And so even if she had learned to read using her left hemisphere earlier on, she still had the capacity and plasticity for this word area to emerge in the right hemisphere. So that's exciting, sort of gives us hope. The oldest, as you all know, the older the child is, the less the plasticity. Well, we don't know exactly. I wish we could say it's like it must happen before this age, otherwise this happens, or you need to get your kid to surgery at this age. We don't know the actual ages, and we hope we will. If we have enough people who participate at some point, we will be able to say, you know, this is what happens if the surgery is really early, and then if the surgery is, you know, in teenagers, and then if it's later, these are the outcomes. It's unknown. We don't have the answer. But I do think that we have some evidence that there is plasticity in older kids where we didn't think that that was true before.
And
Not everyone.
And I just also wanted to add to that, another very kind of interesting thing that we want to look more into and think about is, you know, you can think of something like Rasmussen's encephalitis, where, you know, that's damaging the whole left hemisphere before they even go to surgery. It's possible that, you know, forget about the surgery, maybe the right hemisphere was already starting to take over function before the surgery.
That's what I was going to say. We've demonstrated that with other skills, right?
Right.
That had already probably moved.
That's correct.
Right, and we
We're living somewhere else in the brain, and maybe that's with CBI that's happening.
Yeah.
Have you done any scans on kids that have CBI? Where you could show that maybe some of this recognition is already moved?
So we have now, in these longitudinal studies, begun the scans even before the kids go to surgery, so that we can understand whether or not, even prior to surgery, because the epileptogenic tissue is already malfunctioning, there is already movement of, for example, the word area to the right hemisphere. So we have some data.
Because that's generally an important thing. I mean, you know, you would want to map language or at least have an idea.
Yep.
And one would think if you're moving the whole hemisphere,
Yep, absolutely
the
so.
visual side is the same or similar probably.
So we are doing that. Next BRP, I'm making lots of promises here. Next BRP, I hope to show you some of those data.
There are 2 families here I know that have, I'm sorry, but, like my daughter has TBA, right? And
Yeah.
It's anatomical, it's not disconnect. She doesn't really qualify for your research that you're doing here. At least that's what I was told. But I do know that there's 2 families here that are in my position, and I would suggest, why not? Our goal is to attract more families and couple of families. Is there any value in that to you on the research side?
So my response is absolutely yes. So we've tried to sort of, because we are venturing into these new areas here at BRP, we've sort of tried to restrict a little bit the participants to those who we know have damage to these particular areas, or those have been resected.
Right.
Are we interested in disconnections? Are we interested in TBI?
I would submit to you that the kids with TPO have pretty much the same kind of damage to the visual tract as those with dyspraxia.
I think you're right.
Yeah,
I mean,
demonstrated it in
we know that. I was just going to say, we know this. I already showed it to you. I invite you to come to Pittsburgh. We'd be happy to have you.
How hot is it in the summer 2024?
Yeah, right. You could come to Pittsburgh and we could run the imaging study during BRP.
It's cooler than Houston.
Yeah, it's cooler than Houston. So there you go. The other thing that I will add is we also now have started collecting data on visual recognition in children who have SEEG, so intracranial depth electrodes, prior to surgery. And so this gives us yet another way to begin to understand and sort of piece together what is happening in the areas that are epileptogenic, and how is visual function being affected. And we can look like in parts of the brain that we can't easily see using MRI, and with resolution that we can't really get with MRI. Sean, you had a question.
I did. I was wondering what, after surgery, what happens, and if you're doing any research into the brain matter that's removed.
Oh, so let me just be sure that I understand the question, that I've got it right. You're asking if the gray matter has been removed from one hemisphere, basically they've had a hemispherectomy,
Right.
let's just say. Are you asking me what happens on that side?
Well, more, is there any research into the actual physical material?
I see. There's a long history of neurosurgeons trying to think about ways to deal with the cavity that's left after the hemispherectomy or after the surgical resection. As Monica reminded me, it used to be the case that surgeons would put ping pong balls on that side to sort of take up that cavity. That doesn't happen anymore. It's just a bad idea to have ping pong balls in your head. Yeah, it's like, just don't. Just like don't. And so that part of the brain really fills up with fluid, the cerebrospinal fluid that is typically being produced in the brain. And so that's the current state of the art. There are pros and cons to that. The brain, the remaining hemisphere, can sometimes move, so you get these midline shifts, sort of migrates a little bit further over. The use of hemispherectomy more commonly these days is so in hemispherectomy, there's a little bit of tissue left in the front, a little bit of tissue left in the back. It's to try and prevent it's like an anchor, to prevent the remaining hemisphere from swinging over. So, I'm sure you've heard so much about hydrocephalus and shunts and the remaining cavity. There is a lot of work going on. It's way beyond my scope, and not exactly what I do, but I think people are working hard to try and improve that part of the surgery as well. Go ahead.
I just first want to say thank you for the work that you do. I have a 6-year-old who was a stroke at birth and hysterectomy at 19 months, and we just look at him and go, how?
Yes.
How do you function?
Yes.
How do you do the things that you do? It's fascinating.
I know.
And the things that we see in terms of his sort of vision and language development and recognition, and now he's learning how to read, and we actually sort of suspect he may have some level of prosopagnosia, and we're trying to. So I'm curious how you bridge the gap between what you're seeing and how we look at a practical application of how do I help him harness what he has. I can see him already innately using strategies to compensate, to work around things, to build some of those skills. And I'm just curious if you guys work with other people, on the practical side, the application of it, and how we can help build those pathways that you're talking about.
Great. So first let me respond to your comment when you say, how. I think the how is kind of what drives both Michael and me. There is something really powerful about watching these kids develop and go on to develop skills that you might not even have predicted. And from a scientific point of view, it's staggering for me. It's inspiring for me. We are both have a strong clinical interest. We want to translate the work that we're doing so that it can be applicable in terms of enhancing the visual function. We speak to clinicians a lot, physiotherapists and occupational therapists. My initial training was actually as a speech therapist, a speech pathologist, and I worked for many years as a clinician before I decided to go back to school and take a more scientific path. And it was because I want you to understand, what is the brain doing? If I understand what it's doing, maybe I can make a better intervention. I can customize the intervention. So I think we're still in the process of that translation. But I think there are therapeutic techniques. And OTs and physical therapists know these techniques and are already beginning to use some of them. I sort of hold out for the sort of long-term promise that the science is going to be well translated to the clinical work, and we'll be able to kind of capitalize on what we've learned in order to enhance function.
And I would add that, like, the key thing I think that's really important is, we know a lot of these therapies, from occupational therapy to if you're working with motor things, physical therapy, they're effective in a lot of ways. And, here we're looking at like images of the brain, and we're, looking at like little, you see the little pixels and how they change over time, et cetera, et cetera. And I think, something that we're both interested in is in the long term trying to connect those dots. So, I have a personal interest in the long term understanding, okay, so we have all this knowledge about how the brain just changes on its own over time after surgery, et cetera, but okay, now my kid is getting this intervention, that intervention, this intervention, and then how do we compare kids who have this intervention to, kids who aren't getting that intervention? Is there a difference? Are we seeing kind of this 80% in some kids and then closer to 90% in others? We don't have that data yet, but it's, as Dr. Behrmann said, it's really important to understand kind of the baseline, and then we can, hopefully add the intervention piece on top of it. And that's something that I'm, really excited to think about and do.
Thank you. Question in the corner. Oh, you're next. Sorry.
Can I interject real quick? If your kids are in kid camp, you need to get them. How young are you want to start this? I think we spoke to you like right after we had signed in, and our son is 5. It wasn't really enough at this time.
So the question is, how early can you bring your kids to participate in the studies? We are happy to take 5-year-olds, and it actually depends a lot on the kid. Like there are some 4-year-olds who are really doing well. We had a kid here, a 5-year-old this morning, who actually did everything. It was amazing. Some 5-year-olds can, some 5-year-olds can't, some 4-year-olds can. We happy to try?
Yeah, I think that he could do it. He was all excited about it.
So if you want to talk to us afterwards, we can see whether we can try and find. We have some open slots.
Yeah, and I'll add, anybody who wants to sign up, like, find me after this, because we have some slots left for tomorrow, but I'm the kind of the gatekeeper of getting people signed up. If there is a link going around, don't use that link anymore. You gotta find me. I can give out my cell. I can give out my email. Please get in touch with me to try to get you guys scheduled in.
And actually, Nicole wants to know where she's supposed to go, and you've got the schedule.
Okay, right now.
Go ahead.
So I had a question regarding depth perception.
I can check.
Going back to the example that Michael gave of the picture at airport.
Yeah.
And about your vision cut.
Yeah.
In terms
Has
of,
anybody contacted you?
You see faces and words.
Yeah.
And they're in different planes.
Just give me 1 second.
And looking at the mapping talks, faces are, although they're 3-dimensional, they're somewhat 2-dimensional, as opposed to places and objects, which are much more 3-dimensional. I'd like to know a little bit more about if your studies looked into the differences in terms of that, and what we can learn from that.
It's a great question. I need to be sure that you understand sort of what it takes to perceive depth. Okay. So in order to perceive depth, you basically have to have 2 eyes. Because this eye sort of sees this much, and this eye sees this much. And between the 2 of them, they go, oh yeah, like that is this far away. You've got like 2 measurements of the distance, and that allows you to see depth. If you've only got 1 eye, you can try for yourself, if you've only got 1 eye, things kind of look more flat. You don't have this projection. If you've only got 1 hemisphere, you've still got 2 eyes. That 1 hemisphere will take the signals from both eyes. And so you will still have depth perception, even in a single hemisphere.
Assuming the eyes are working together.
Yeah, I'm
Right,
assuming the eyes are working
Right,
together.
Right.
Yeah.
And then I think you
Absolutely.
So a lot of kids have exotropia.
Yeah.
Sure. Yes.
Yeah, I've been trying to stress the fact that you have to have your eyes sort of
Aligned.
synced up.
They have to be
They have to be aligned. So just to be clear, one hemisphere can actually be enough for depth perception, assuming the eyes are aligned.
I meant in your studies,
In our studies,
were you
okay.
able to kind of,
Yeah.
I'm gonna say,
Alright. Yeah. So
Six.
in the studies that we've done, the kids are lying in the magnet or doing the behavioral study in front of the computer screen. These are all just two-dimensional. It's just all flat. When we show things in the magnet, it's really like you watch a movie. It's just on a flat screen.
Can you send me a
We
text?
would love to be able,
I'll have the answer that
and
we
we
don't have that
will,
information right now.
use virtual reality in the magnet and in our studies to examine what happens when you really do feel like you're immersed in a 3D environment. And what is the visual system doing under those circumstances? In some ways, that's an even more complex question than what we're even trying to answer right now. But it's a really important question because we want to know about performance in the real world, sort of naturalistically. Sort of like why we show this movie, because it's a bit more of an immersive experience. It's like the closest we can get to about naturalistic experience when you're lying in a magnet with your head bolted down. It's a great question. We've got a lot of work to do. As you know, the day is young in this field.
So
Thank
my
you.
next question was going to speak kind of like straight right to that, in that, you know, with normally developing kids, if you develop, you know, the exotropia or the eyes that aren't aligned, right?
Yeah.
The prevailing wisdom is that to prevent the amblyopia, you've got to address that and
Yeah.
straighten the eyes out. But a lot of these kids, like mine, they've got this massive field cut, and you can see one eye is moving, and you can understand over time they're trying to naturally expand their field. However, that's going to come to the detriment of
That's right.
his
They're lying.
depth perception. But yet, you will see adults walking around with eyes that are really messed up, but they're doing just fine. Like, how is that possible? Because you know they're not projecting on the same, with
That's right.
foveal vision, they're not projecting the same on the
Yeah,
same spot.
that's right. So when the eyes are out of alignment, it's like your left eye sees sort of one bit, the right eye sees something completely different. So my left eye may be seeing this, but my right eye may be seeing Michael. And so I can't actually bring those into alignment. Like that doesn't match, that doesn't work. Whereas if our eyes are aligned, my left, you know, I see this with my left eye and this with my right eye, it sort of becomes seamlessly integrated. We in fact don't even know what each eye is seeing. We don't feel, we are not monitoring what each eye is doing. It's sort of seamless and it gets stitched together. But if the eyes are separated, there is really no stitching together, and there is a field cut on top of it. So this is hard.
It's hard. Like, what do you do as a parent? Do we work hard to try to fix that with the younger kids? Anyway, with the younger kids, you can patch and all that sort of thing, or not, or just let it go. And just,
So I know when I should say I don't know the answer, and this is one time that I don't know the answer.
We talked about it in 2019 at that session.
That's right.
There's no consensus, that's the problem.
That's right.
But I will say, I believe, this is my personal opinion, that most of the neuro-ophthalmologists are saying you have to correct surgically. Otherwise, with the patching really isn't working with the kids. If you don't, then you lose that depth perception. Then you've got a double win. So no depth perception and hanging.
exactly. Right.
Yeah.
So
My son has, by the way, so because of the challenge he's had with hydrocephalus, he had the exotropia corrected, then we had multiple shunt failures. He's had so much damage to his ocular motor control that one eye has lost vision completely because it can't move anymore. So if you're welcome to look at him, he cannot move his one eye out, he cannot move his eyes up anymore because of all that. The result then became he's probably lost all his vision in his left eye. So you really need to make sure you're
you can have a sense for yourself of what it's like if you've only got one eye. If you just close, so like my left eye can see all the way from here, actually almost to Monica. I can't see beyond. So one eye can give you information both on the left and on the right. But what you really want is you want both eyes because they have these slightly different samples and it makes the world look seamless and meaningfully integrated. And these are really, really hard challenges and multiple challenges simultaneously. Amblyopia, extratopia, together with visual field cuts, and other kinds of ophthalmic disorders as well that co-occur.
When it comes to reading, do you see benefit with prism glasses?
Yeah. I was going to answer Ken about prism glasses, and I decided to hold off on that because I don't know a ton of the literature that has evaluated prism glasses. I have a friend who is a neurologist in London, and he has actually built a system for prism glasses, mostly for adults who've had strokes.